Hi, everyone. My name is Chelsea Madden, and I'm a scientific solutions consultant at Benchling. And today I will be talking to you about how Benchling can advance cultural foods through digitalization and our lab automation software. So, as you all probably know, the cultured foods industry is undergoing a significant transformation. It's still a fairly young, new industry and therefore is constantly evolving and growing. Some specifics that we've seen in the industry are that there's more novel strains being discovered. So some of these things lead to more difficulties, a little bit more challenges in managing all of this. So when there's more novel strains, there's more things to keep track of from your day to day. There's also increasing complexity of your scale up data due to a number of different tests that are being performed increasing. And there's also greater sophistication of bench scale technology being used in the lab. There's lab automation software and instruments that are being used that makes the data capture a little bit more complex. So Benchling can help to manage your full R&D process and all of these challenges that we're seeing in the industry. To talk a little bit about the Benchling platform, is a unified cloud-based R&D informatics platform that allows you to manage everything from your experiments through notebook entries to managing in silico design with molecular biology tools, to sample management and overall managing your R&D process flow. The system is configurable and is able to be flexible to manage specific R&D functions, but it does not require any code to do so. So it's all point and click configuration to be able to achieve this. It is also, as I said, a cloud platform, so access via the cloud, and the platform is open so that you're able to integrate and connect to any other systems or instruments that you need to through open APIs to ultimately make your R&D process more efficient. We have a number of different current customers in the industry that we're really proud to partner with. And what's been really great about working with these customers in the strain engineering and fermentation space is that we've been able to grow and scale with these companies as they grow and scale and as the industry grows as well. And this has really led to Benchling developing pretty great domain expertise in this strain engineering and fermentation space. We're excited to continue to work with these customers and gain new customers and partner with them as the industry continues to grow. So this is kind of the typical workflow that we're seeing at a high level in the cultured food space. So everything from, starting with the plasmid design all the way down through analytical and food testing. So what I want to talk about is how Benchling can help with each stage of this process. So we'll look at it through the lens of a couple different personas, starting out with the strain engineer who's really managing plasmid design and strain engineering. So from a strain engineer's perspective, they could be designing thousands of plasmids and keeping up with the throughput as well as keeping track of all of the different parts and strains can be a challenge. So let's go ahead and jump into Benchling and see how we can manage this. So I've opened up to the Benchling application here. You can see that the way I navigate through the system are my icons on the side. And as a strain engineer, maybe the first thing I want to do when I jump into the system is to search for all of my strains that I want to work with. So I can do so by using Benchling's global search. So let's go ahead and I can see that I can use a simple text-based search, or I can also use a filtered search. So first I'll search on all of my strains and it will then pull up all of my strains managed in the system and show some metadata associated with them. I can also do a more filtered search where maybe I want to look at strains with a particular gene or genes included. So I can select my field and say I want to see all of my strains where it includes these particular genes. A lot of flexibility here with the search in Benchling to make it easy and quick to find exactly what I'm looking for. So now I've surfaced 3 of these strains which are expressing these particular genes. And I can see here that Benchling makes it easy to track lineage of everything that I'm tracking in the system. So for the strains, I can see the host strain that was transfected. I can also see the plasmid, of course, those genes. I can also access all of these, so I can see that they are linked chips. So if I want to take a look at my plasmid in detail, so let's say if I want to focus on this particular plasmid for this strain, I could go ahead and click into that, and I can see all of the details of this particular plasmid. I can see here that Benchling is sequence aware and has molecular biology specific tools that I can use to do my in silico design. I can also see that I can manage all of my metadata associated with my plasmid here. So I can see, for example, that it's associated with a particular project. I can also see the links into the lineage of the backbone plasmid and my gene inserts. And Benchling can also surface some of these parts as well on its own. As I said, it's sequence aware, so I can autofill these things and it will recognize things like promoter and resistance in a plasmid sequence. So one really nice tool, especially for this use case of the strain engineer, is the ability to bulk assemble DNA. So it can be a very manual process to have to do an assembly of a plasmid one by one. So Benchling gives the ability to bulk assemble DNA. So I've accessed our bulk assembly tool here, where I can see I have selected 3 plasmid backbones that I want to use for this assembly. And I've also selected 3 different gene inserts that I want to assemble with those backbones. And what I can do is use this tool to assemble those. And Benchling will automatically create these plasmid constructs. And if I open the folder that I've saved them to, I can now see all of my newly created plasmids here managed directly in Benchling, and I can do all of the work and experimentation that I need to with these particular plasmids. So you can see how Benchling helps the strain engineer persona to manage a lot of different strains, plasmids, as well as manage the in silico design of those plasmids. So let's go ahead and move into the fermentation engineer's perspective and persona, where they're managing the small scale culture and culture scale up. And from this fermentation engineer perspective, they could have a ton of online and offline data that they're managing and tracking, which are stored in different places and can be difficult to kind of consolidate in one place and understand strain performance based off of all of that data living in disconnected places. So let's go ahead and jump back into Benchling to take a look at how we can manage that. So what I can do here is I can have templatized entries for my experiments to really standardize the work that I'm doing in the lab. So for fermentation, I could have a specific templated entry to walk me through my experiment. So I've opened one up here that I've already created, and you can see now in this lab notebook entry, the ability for me to use this to walk through my scientific process. So I can have things like images, structured, unstructured tables included within my experiment entry. So as the fermentation engineer, the first thing I need to do is create my fermentation batch here. So I can do so by selecting the strain and the seed culture that I want to create this from, as well as the media that I'm using. And once I go ahead and submit that, it creates this new fermentation batch, and it automatically is able to surface the genetic construct of this new batch based off of the makeup of the strain. So again, Benchling can pull that information and that lineage information. I then want to go ahead and collect a number of time point samples for this new fermentation batch. So I can go ahead and create those again directly from my batch here for all of my time points, and I can see those within this structured table in Benchling. Now I may have offline data that I'm capturing for all of these time points. Maybe I'm capturing OD and pH levels. So I can do so within a Benchling results table. I can either manually enter this data into my table, or I also have the ability to upload from a spreadsheet. So I can easily enter and upload that data that I'm capturing offline and have it directly related to these time point samples for my fermentation batch. So I have all of that information in one place. Also, as a fermentation engineer, obviously, it's important to be able to visualize all of the data that I'm collecting so that I can really understand that stream performance. So I can see here, I have an example fermentation dashboard within Benchling's Insights tool that I can use to collect that data and visualize my performance here. So for example, maybe I want to look at product titer for a particular fermentation batch. I can see those across different batches. I can also view information in a table structure here, so I can see my fermentation batch and then downstream information as well. So as I continue through the process, the R&D process here, I'm able to collect all of that data and have it linked. I can also see additional data around OD growth curve that I'm collecting and food scoring. So a lot of different information I can collect from the data that I'm capturing, whether online or offline in Benchling here. So now as a fermentation engineer, I want to go ahead and send a request to the analytical team to be able to request that time point sample assay. So here I can see what I need to do first is to transfer my fermentation batch time point samples into a plate. So I can use a structured table to do that in Benchling. So I've transferred all of my new fermentation time point samples into this fermentation samples plate. And if I click into that, I'm able to visualize that in Benchling as well. So I can see each of my samples and their positions, and I can see those linked in as well. So what I want to do is go ahead and send a request to the analytical team, as I said. So we can create a request from these samples, and we want to create a time point sample request. I can add information around assign to. For the purpose of the demo, I'm going to go ahead and enter myself. I can give, date requested by, project as well that it's related to, the request type, and then I can select exactly what sort of assay or test I want done to these particular samples. So for this, we'll go ahead and say we want to request an HPLC product titer. I can toggle all those for the samples I want and create, and now the analytical team will be notified that they've been sent that request and will be able to go in and fulfill it. So we've seen how we can manage that online and offline data and visualize that as a fermentation engineer. So lastly, we're going to go ahead and take a look at how the analytical scientists can use Benchling to manage samples from multiple runs, as well as help them to return assay results more quickly. So again, we're jumping back in here to Benchling. I can see that now I have this time point sample request sent from the fermentation engineer. As the individual on the analytical team, I can go ahead and execute now, and I can do so in a template. So again, standardizing how I am completing all of these requests and doing all of my science here. I can create that from template. I can see that it surfaces all of my time point samples that I am doing my HPLC run on. And I can see now that I'm able to use Benchling to automate this run. So what Benchling can do is generate an instruction file that it can feed to an instrument, in this case, the HPLC. We've done it with multiple instruments. Then the instrument can return a results file, which can automatically be loaded back into Benchling via API, or it can be manually loaded into Benchling and can be automatically associated with those time point samples. So let's go ahead and create our run here. I can select my sample plate. I can select the solvent I want to use for this run, as well as the flow rate. So specify my metadata for the run. I can create. I can generate my input file that's going to be fed to the HPLC. And then in the background, once the HPLC has done the run and created that output file, that can automatically be loaded into Benchling. So I can process that result. And I can see now that for my run in Benchling, it surfaces all of the results from the HPLC run associated to my fermentation batch time point samples. So I can now see the product titer results for each of these, and that will be accessible on Benchling now for all of those samples. Last thing I want to show here is that we looked at a dashboard for the fermentation information. I can also have more operational dashboards to show me information around requests. So as the analytical team, I can view operational information around requests per week. I can also keep track of my request statuses and all of that type of information as well. So insights can really be used for that purpose of operational data capture as well. So we've kind of gone through the typical challenges that we've seen in the cultured food space, from the strain engineer to the being able to embed sequence level intelligence and track samples, to the fermentation engineer to unify online and offline data, to the analytical food scientist to really improve the productivity of the analytical team and help them manage all of the different samples and assays that they need to. So if there are any further questions, please let me know. Again, here's my information, and we have a few minutes here to go through some questions. Thank you.